In the last video, you learned about PCR and the centrifuge. Now we will talk about two relatively new tools in synthetic biology, RNA interference and CRISPR. Normally in our cells, DNA is transcribed into mRNA in the nucleus. Then the mRNA is exported to the cytoplasm, where it binds to a ribosome and is translated to produce a protein. RNA interference, or RNAi, is found naturally in many eukaryotic cells and can be modified by researchers. The goal of RNA interference is to knock down a gene by degrading or sequestering the mRNA. Due to the fact that it targets mRNA and doesn't alter the actual DNA sequence, RNAi is a good choice for making temporary controlled changes in cells. Two types of small molecules of RNA, microRNAs and small interfering RNAs, are crucial to the process of RNA interference. miRNA precursors are synthesized by the cell itself as long, hairpin-shaped, double-stranded RNA molecules. siRNA, on the other hand, is produced from double-stranded RNA originating from outside sources, such as from viruses. Both miRNA and siRNA precursors are cleaved by the enzyme dicer into short 20 to 25 base pair long, double-stranded fragments. The fragments then complex with the protein argonaute and one of the strands of RNA is discarded. This complex may further bind with other proteins, forming the RNA-induced silencing complex, or RISC. RISC uses a single strand of RNA to bind to complementary sequences on mRNA in the cytoplasm, and then the complex either cleaves the mRNA or blocks translation. In the lab, researchers can synthesize siRNA molecules with specific sequences in order to silence the expression of particular genes. This allows them to evaluate the function of unknown genes, and RNAi can also have therapeutic benefits as it can help to silence genes that may be overreactive in certain diseases. Next, CRISPR-Cas9 is a gene editing technology that can be used to correct errors in the genome, potentially allowing us to cure many genetic disorders. CRISPR was originally found in bacteria as an antiviral mechanism. Bacteria would integrate viral DNA as spacers between repetitive sequences in the CRISPR locus. These spacers would be transcribed into a special type of RNA known as CRISPR RNA or crRNA, and then would complex with tracer RNA, which is a constant stem loop shaped RNA, and two of these RNAs would complex with a Cas protein. Upon future infection, the Cas protein could make a site-specific cut in the viral double-stranded DNA based on the crRNA sequence. Researchers are now able to utilize this process by synthesizing guide RNA, a combination of CRISPR RNA and tracer RNA that can target the Cas9 nuclease to the desired site for editing. After the cut is made, DNA repair may occur by two mechanisms. First, the two ends may simply rejoin, known as non-homologous end joining, leading to a small random insertion or deletion. On the other hand, in homology-directed repair, a homologous sequence of DNA may be used as a template to repair the cut. Researchers may deliver homologous sequences containing the desired changes along with the CRISPR-Cas9 system so that when the DNA is repaired, it takes up the changes from the homologous sequence, permanently eliminating the mutation from the organism. That's it for this video on RNA interference and CRISPR. In the next video, you will learn about bioethics. Thanks for watching.